Good afternoon. I'm Gary Rabin, Chairman and CEO of Advanced Cell Technology. I have a 25-minute speech to give in 13 minutes, so I will kind of skip ahead now. I haven't any idea how to move this forward. Okay. All right. Okay, beautiful. So Advanced Cell Technology is a publicly traded biotechnology company, ticker ACTC. We've got a market cap of about $165 million, headquartered in Santa Monica, California. And the company's principal manufacturing facilities and research lab are in Marlborough, Massachusetts, in a little bit of the eye of the storm today. The company uses a variety of pluripotent stem cells to create our stem cell therapies. And one of the main distinctions between Advanced Cell and so many of the other cell therapy products that are going out there is that we are really creating an off-the-shelf, fully scalable, allogeneic source of tissue for our clinical trial projects. We use human embryonic stem cells, but the way that we culture and derive these embryonic stem cells is totally different from the conventional methodology for creating embryonic stem cells. Normally, embryonic stem cells are cultured through the removal of the inner cell mass of an embryo. And when you do that, you destroy the embryo. We do not do that. We take a much earlier stage embryo when it's at the 8-cell morula stage, and using a pipette, we pick out a single cell. So we do a single cell biopsy, removing a single blastomere from that embryo, which doesn't change the fate of or harm the embryo in any way. We've got 12 embryonic stem cell lines, and in the case of 11 of the 12, the embryos were preserved, remained viable, frozen down, and so that gives us an important legal, moral, ethical, religious, political advantage over the traditional method for creating embryonic stem cell technologies. We are also looking at some interesting things in IPS, and we actually have one of the earliest controlling filings in the use of OCT4. I want to talk about our lead clinical program right now. We are involved in a kind of cell, creating a kind of cell from embryonic stem cells called the retinal pigment epithelium. And the retinal pigment epithelium sit at the back of the macula, and they provide a whole variety of trophic factors and phagocytosis to the photoreceptor layer. What happens is, as the RPE layer ages and goes through a decline and eventually cell apoptosis, there's a corresponding decline in that photoreceptor layer. Now, that is because the RPE cells recycle vitamin A and detoxify the photoreceptor layer, but they also provide another important function. They secrete and maintain what's called the Bruch's membrane. The Bruch's membrane is the natural anti-angiogenic layer between the front and the back of the retina. So when that Bruch's membrane begins to degrade as a result of, again, the failure of these RPE cells, what you get is the leaching into the back of the eye of the choroidal capillary infrastructure in the back of the eye. That's called choroidal neovascularization, and that leads to a disease called wet AMD. Now, there are several therapies available for wet AMD. There are anti-angiogenic, basically drying out drugs that help treat these things. But there's no cure for wet AMD. But we're actually going after a different segment of the AMD market. We're going after the dry AMD market. Dry AMD represents 90% of the total AMD market, and wet is 10%. And in fact, one of the interesting things about wet AMD is that in almost all instances, in about 85% of instances, the precursor to wet AMD is dry AMD. Sometimes it's not. Sometimes it's from retinal distension and all different other sorts of potential causes, but typically it's caused as an early stage through dry AMD. So 90% of the AMD market has no treatment or therapy available to it of any kind. You've got one of the largest unmet medical needs in the world. And like I said earlier, we are pursuing this therapy here that is very scalable, very repeatable, off the shelf. We essentially, in our 1,000-square-foot GMP manufacturing facility in Marlborough, Massachusetts, using the existing shifts that we do, single-day shift only, we could make about 500,000 doses a year of this RPE therapy. And that's in a tiny little 1,000-square-foot space. Now, like I said, we're going after the dry AMD market. And right now, we've got a clinical trial ongoing that's treating this sort of late-stage part of the patient population. These patients have virtually no photoreceptor function in the central field of vision, or fovea. But that's not the market, obviously, that we're going to chase after. we are doing something different than all of the other AMD drugs that are in the clinical trials right now. Those drugs are sort of designed to sort of slow down the process of decline. We're doing something completely different. We're injecting dissociated RPE cells, and what we're trying to do is recapitulate the infrastructure in the back of the eye. And what we've seen in our animal studies, and in fact in some of our human models so far, we've seen this actually working. And so what this suggests to us is that as we begin to treat intermediate and early stage patients, we're going to really see very dramatic changes in visual acuity and a complete slowing down or stopping of the decline of this disease. In the preclinical trial material, we saw that when we injected these dissociated cells in the ELOV mouse, which is a Stargardt's macular dystrophy mouse model, we saw that these cells injected and biodistributed, laid down, and formed this monolayer on the mouse Brooks membrane. You can see the fluorescent cells there in that top right slide. The fluorescent cells are the human cells laying down adjacent to the mouse RPE. Now that is, to us, a very powerful signal for what we could expect to see in the human models. On the bottom here, the left slide there shows the control eye of the treated RCS rat. That's the dry AMD rat model. And the control animal, that's the photoreceptor layer. And there you can see it's a highly dissociated photoreceptor layer, lots of cell apoptosis. That animal is virtually blind in that eye. Same animal, treated eye, on the right side there. That animal has a photoreceptor layer, 5 to 7 cell widths in thickness. That animal's got about 80% the visual acuity of a healthy rat. So in our animal studies, we saw really impressive results. Now we've got 3 trials ongoing worldwide. We've got a dry AMD trial in the US, and then we've got in the US and the UK, one each of Stargardt's macular dystrophy. That's a juvenile onset form of macular degeneration. It's actually born out of a genetic defect, but the end result is the same. Toxins build up, kills off the RPE layer, which kills off the photoreceptor layer. So we've got these trials ongoing right now. We've treated, as of today, 13 total patients, and within the next couple weeks, we'll be up to 16. Each of the 3 trials is set up in 4 cohorts. It's an escalating dosage trial. We've treated the 50,000 cell dosage, and we've treated most of the 100,000 cell dosage in these trials. And then we'll escalate to 150,000 and potentially 200,000. Our participating retinal surgeons in this are really a valuable part of this. We have 4 of the 5 top eye hospitals in the United States as our clinical trial investigators. Our lead clinical investigator is Steve Schwartz, who's the head of the Retina division at Jules Stein Eye Institute. We also have Wills Eye Institute in Philadelphia, Bascom Palmer at the University of Miami, which is the #1 eye hospital, and Mass Eye Near at Harvard. And then in the UK trial, we've got Jim Bainbridge, who is the chair of the retinal studies program at Moorfields Eye Hospital, which is undoubtedly the #1 eye hospital in Europe and possibly in the world. The surgery itself is relatively straightforward. Because I only have 12 minutes, I'm going to skip over it here, but it's about a 90-second surgery. It is an outpatient surgery. The patient goes in, local anesthesia, there is a vitrectomy, and then the cells are injected into a bleb, and then the patient is kept prone for about 5 or 6 hours afterwards to allow the cells to settle. In January of this year, we published a very interesting report in the Lancet, which showed the anatomical evidence of the RPE cells surviving and engrafting and increasing pigmentation within the bed of the transplant, within this bleb. And then we also saw in both of those 2 patients that were the topics of the Lancet article, as well as most of our subsequent patients, improvements in their visual acuity. These changes in visual acuity surprised us, because we thought that given the degree of atrophy in the central field of vision in these patients, we didn't think that there would be any photoreceptor function to rescue. You're talking about patients that are legally blind and in many ways, for all practical purposes, totally blind. Hand motion type patients are 2800 vision. So you're talking about very, very low vision patients with massive areas of atrophy within the fovea. I want to show some new pictures here that are pretty relevant, that we haven't shown before today. The baseline, this is actually from one of the UK patients. You can see the injection site there, and then you can see as month 1 and then month 2, you see increasing degrees of pigmentation. And that shows the persistence and the survival of these cells. So this was something very, very gratifying to us. And again, this patient has had some improved vision, some improved visual acuity as measured on the ETDRS chart, but also more importantly, much greater recognition of color, contrast, and light. So let me skip ahead now a little bit. So in terms of upcoming milestones for these trials, we've got a total of 36 patients to treat in these 3 phase 1 trials, 12, 12, and 12. But what we really want to do now, and we just actually last weekend had a kind of a getaway with our clinical investigators and the members of our independent ophthalmic advisory board. And what we really wanted to get to was how is the best way for us to go about starting to treat earlier stage patients? Should we hop into a phase 2A trial and power it with more data and see if we can start looking at what happens when we treat 2100 or 2080 patients? Or should we just go in and try to change the protocol to include earlier stage patients in our phase 1 trial? We've actually chosen the latter. So we're going to be going to the FDA asking for some changes to our protocol that will allow us to treat earlier stage patients. And we think that this will really be something that will be very powerful. It'll be a very powerful tool when we show this to, you know, the big pharma type companies. That's been the one thing that they've asked for is, this looks great in these blind patients, but what happens when you treat people that actually have some vision? So that's an important sort of threshold for us to really be able to meet. We want to, of course, define what the efficacy endpoints and the visual acuity criteria will be for these patients, and of course, as we get into the phase 2 trial. And of course, we also want to sort of start simplifying the shipping and the cell prep to enhance scaled distribution of this. We have very significant intellectual property around this. We've got patents around not only how the cells are cultured, but also the specific characteristics of the cells themselves. So really a lot of important filings going on sort of constantly around the clinical procedures and so forth, really creating quite a thicket around our intellectual property. And obviously, talking about this as a massive market size, if we just had 1% penetration in this market, and we had $5,000 per dosage, you'd be talking about a $5 billion market. I mean, that's a simply enormous market potential opportunity for us here. We've got some other programs in the eye space. We've created sheets of corneal endothelium. We've identified a new kind of previously unwritten in the literature form of photoreceptor progenitor cell that can be a rod or cone, but it's effectively a postmitotic cell. So very interesting opportunities for us here in the eye space. And then another area that we're really starting to focus keenly on now is mesenchymal stem cells. Now, mesenchymal stem cells, as you are probably all very aware, there are quite literally hundreds of MSC trials going on worldwide. All of those trials have one thing in common. They use some adult stem cell source for the MSCs. And what we've found in both the factors expressed and the replicative senescence issues that are created by using these adult stem cell tissue derived MSCs gives us a huge differentiator when we use hESC-derived cells. And we'll be talking more about what we're seeing in our preclinical information here in the coming months. Just wanted to flash forward just a little bit here and talk about the company itself. We've really turned around as a company in the last year and a half since I took over the company. I've redone the entire board of directors. We've added world-class scientists, very well-known people in sort of taking companies from early stage into commercialization and into partnership. And so having this kind of board of directors is really very important to us. And then just a couple of weeks ago, we put in place a new $35 million at-the-market financing commitment in place. That really makes a difference for us. That funds the company effectively into 2015. So we ended the third quarter with a combination of cash on hand and availability under this financing commitment of about $42 million, $16 million annual burn rate. So like I said, that gets us through a good part of 2015. Thanks very much.